Hi, in this video I'm gonna talk about how to select a library and things you can think about. The first step in any project, right? I'm gonna remove myself here. So this refers to this area, right? Where 8 spacer libraries are embedded, 4 human, 4 mouse. And then there's also a user uploaded version, which I'll talk about in a different video. So when you select a library here, you'll see which publication it comes from, links, other information that could be useful. The Brie and the Brunello is coming from this 2016 paper, and these are the libraries. The Brie and the Brunello ones are the ones that we use the most. I think traditionally in the last few years, I think they've been the most used ones. I'm not gonna take poison on that, but it's commonly used together with a couple of other ones at least. The Gecko libraries are a little bit older. I think they were used a lot sort of in the beginning when all this sort of CRISPR screen stuff started, and nowadays less used. I don't want to have strong opinions about this, but I would argue that the Brie and Brunello library probably has a more advanced on-target calculation score. Then we have the Gattinara and the Goda libraries. Gattinara and the Goda. That's coming from this publication. John Donch as the senior author, which of course he has been very instrumental in a lot of these, accelerating this field and developing this field, and has designed a lot of different CRISPR screen libraries or spacer libraries, including this one. I'm not sure right now if the Gattinara and Goda are different in the algorithms used to design them to the Brie and Brunello. You see he's the first author here. I'm under the impression that they are the same or very similar at least. But I do know at least that the Gattinara and the Goda library contains 2 guide RNAs per gene, while the Brie and Brunello got 4, and these are not overlapping. So if you read this paper, you see that you could use them in different combinations. Sort of, you can combine them, or they're not overlapping at least. And also we included this because this is a more condensed library, and that's a trend, I would say, that people start realizing that you can use smaller libraries and still have good discovery, but using fewer cells, right? And then lastly, we included the VBC library, which we started to use a bit, and it's working really well. And I want to say 2 things about this. One is that this one, in its design algorithm, it wants to target conserved regions. It's trying to get the guides to target conserved regions, conserved over evolution then, and with the idea that this means that these regions that you target are encoding for protein domains that might be very important for the protein function. As you know, it's a common sort of idea about conserved regions. And that will have a potential then a higher chance that if you do anything in that region, even if it's just an insertion or deletion and not the frame shift, that you will be able to generate the loss of functional allele that as they're talking about here. The other thing I want to mention about this library, and this is important, could be very important for you, is that they, while all other libraries typically have 20 nucleotide long spacers, this actually have 18 to 21. And the reason for this is that they want to use an endogenous G. So the U6 promoter that's commonly used to transcribe sgRNAs needs a G to start the transcription. And most libraries don't, or most spacers, when you design them based on the sort of algorithms that are used, don't have a G in position 1. And the trick then is just to add a G. So when you add your adapter sequence that you're going to use for cloning, you make sure that the terminal nucleotide there is a G, and then there's going to be a G that's before the start of the nucleotide. In contrast, the VBC libraries makes sure to use a G. So if it's possible to use an endogenous G, so a G that's part of the sequence, if the spacer is made 18, 19, or 20 base pairs, they're going to use that. And if that's not possible, but the guide or the spacer is still scoring very high in there with the algorithm, they're going to make a 20 base pair guide spacer and add a G before that. So that's the 21 version here. So you can see if you add a number of genes here, so this is human, so let's call it T53, IL4 receptor, this STAT3, common. You'll see that all of them are starting with the G, right? Boom, 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 boom. So there's always going to be a G there. And this has a practical application that I want to highlight. And that is, if you're using this library, you should consider to not add an extra G, which you might do by default. So the adapter sequences we use, and you can and we clone into LentiGuide Puro, typically, you can get those by loading test data here. You'll insert the ones that we use for cloning. You'll see that that one has a G here, right? But if you want to use the VBC, and that makes sense. So here is using the Brunello library, and what you'll find then, and here we're limited to top 2, so that's why you only see 2 guides each here for these. You'll see that one guide here has a G, another one has a G, but most of them don't, right? That's the capital letter. But we make sure by having a G in the adapter sequence that there's going to be a G there. So for the Brunello library, this is, and basically for all libraries, as far as I know, this is how people do this. But if you want to take advantage fully of the VBC and their endogenous G, and I haven't seen any sort of benchmarking if to what extent it matters if you do this or not. But if you want to take advantage of this idea, you would need to remove this terminal G, right? Because otherwise you're going to have a situation looking like this, where all of them are going to have a double G, and of course some of them are going to have more than, as you see here. So whether it matters or not, again, I'm not 100% sure. I haven't seen any, but it makes sense to either remove this G, and then you'll at least not have an extra G before there, or, and none of these things are better or worse, but I'm using them also as a sort of a learning experience for you, or a possibility to show you functionalities. You can also trim one nucleotide in the 5' end. So the first nucleotide in the guide or spacer will be removed, and then you achieve the same thing. So we kept the terminal G in our adapter sequence, but we removed the first nucleotide in the spacer. And I mean, here these are shown by small and large letters, but or lowercase and uppercase, but of course for in the cell this is not going to make a difference, right? This is just for demonstration purpose. I mean, a G is a G, right? And A is a. So that's 2 versions of using it. Trimming it or removing it from here. The trim function, I could just take a minute or half a minute to just mention that you typically won't use this working with this, but, and I'll get to that later, but if you use a user uploaded one, sometimes you get libraries that contains extra sequences that you want to get rid of. Typically you have some libraries that contains a PAM sequence, and then you can remove that, for example. So that's sort of why we implemented this trim function. Yes, so that was the video about selecting libraries. There are different ones. VBC, a little bit to think about there. And with that, I thank you for watching this video and continue doing that. And I'm hoping that your CRISPR screening goes very well. Thank you.